Okay, I want to keep going with our history of DNA. We started back here with Frederick Griffith back in 1928. And remember, Frederick Griffith, he had no clue what the transferring material was, but he at the end said, he was able to prove definitively with this experiment with that heat killed bacteria and the live R bacteria that something got transferred between the bacteria. He didn't know what it was, but something had to. So without a doubt, something's actually getting transferred. Well, then along come these three guys, and the protein was a player in the game, and this newer molecule at the time called DNA, they thought that might have had a hand in it. Well, they did their experiment, and like we said, DNA is the genetic material, not proteins, because when they took the proteins out, everything stopped. When they took, everything continued. When they took the DNA out, everything stopped. So now we know it's the DNA. Well, now we've got these two scientists, Hershey and Chase, and oftentimes in the scientific world, we fast forward 8 years. So now we're at 1952, and so Hershey and Chase, they just want to confirm. They want to run their own test and say, well, just because these three guys said it's DNA, that doesn't necessarily make it so. And so other scientists want to test it and see what their results were. And so Hershey and Chase, remember, I'm not going to ask you for dates, but Hershey and Chase wanted to test it as well. And instead of using bacteria, they used viruses, and they put them in a blender of all things to try to figure their version of it out. So they were using viruses instead. And so here it is up here on top. This is, believe it or not, you guys, that's kind of what a virus looks like. They're kind of freaky science fiction type stuff, but those are viruses that make us sick. And notice what they did is they put little tags on the DNA. And so the DNA that was located in the virus, they put a little tag on it. It says a radioactively labeled. Don't worry about that. We're just going to call it a tag. And so then if you zoom in, well, now you see all these little viruses, and it's attacking a bacteria. And so now you notice that DNA went from being up here, and now it's inside the bacteria. So there's the virus attacking the bacteria. What's being injected? The DNA. So then we've got the empty husks of the viruses, and they put those in a blender. And the DNA from the virus was now found inside the bacteria. So by using markers on the DNA, they were also able to confirm what Avery, McCarty, and McLeod did, and proved that the DNA is the transferring material. And so now again, other scientists are starting to yeah, it's DNA. And so now around this time, scientists are starting to want to figure out, we want to know, what does DNA look like? And there was this huge race. And so all the focus at this time, the late 40s and early 50s, was focused on, the actual structure of DNA. So now we fast forward. There's DNA spelled out. You and I will very seldom see it spelled out, deoxyribonucleic acid. It's stored in the nucleus. That's our genetic information. You got your DNA from your mom and dad, half from mom, half from dad. And then many of you will become parents down the road, and you're going to give half of your DNA to your offspring, and your spouse will give half the DNA, and two halves make a whole, of course. And so that's where that genetic information comes from. And that's where we now know that's why we look like our parents, and siblings look alike, is because DNA is actually transferred from one generation to the next. We now know the structure. It's made up of nucleotides. Way back when we did macromolecules, you guys, we did carbohydrates, proteins, lipids, and nucleic acids. And at that time, I mentioned that we're going to do a whole unit on DNA. Maybe you have forgotten, that's okay. But we're now at that DNA unit, and so here it is. And we talked about that. So the phosphate group. And then it's got the sugar, which is ribose, and then one of 4 nitrogenous bases. And so our DNA, you guys, is made up of millions of these bases repeated over and over and over. And if we go back to our proteins, with our next slide, I talked about proteins. There's 22 amino acids used over and over and over in all living organisms, which is just wild to think about, that the variety of life with the trees and the bushes and the birds and plants and animals and dogs and cats that were all made from the same 22 amino acids. Well, on Earth, we're all made up of DNA, and if you count up the bases for DNA, 1, 2, 3, 4. There's 4 different bases for DNA, and every living organism on our planet is made up of those same 4 bases repeated over and over and over. And this gentleman here, I don't know if it's Irwin or Edwin, I think it's Irwin Chargaff, he figured out that adenine always pairs with thymine, and so we're going to see it written like this: adenine bonds to thymine, A bonds to T, and guanine pairs with cytosine, G bonds to C. We're going to get those pairings down the road, and that's going to kind of become something that we'll have ingrained in our memories. But that's part of the DNA as well. Those 4 bases get repeated over and over, and it looks something. Irwin Chargaff got that right in 1949, so he was studying that stuff at the same time. Well, now here's kind of the idea of DNA. Oops. And so it's called the helix is the scientific way of saying a spiral. And so it's 2 spiral, kind of like spiral staircases bound together. And so here we see that T bonds to A, thymine to adenine, guanine to cytosine, cytosine to guanine, adenine to thymine. So we see that A to T, C to G, and then right here, this is an important piece too. The 2 strands are held together with hydrogen bonds. And that, you guys, that's the code of life. That's what makes us actually. And then the last piece, in terms of the actual structure, these 2, Rosalind Franklin, she was using X-rays. Is that on? Yep, X-rays. She was using what's called X-ray crystallography. So this is Rosalind Franklin, and she was working in one lab in London, and she was taking her X-ray pictures. And unfortunately for Rosalind, she was exposed to a lot of X-rays because of her work. Well, she ended up dying of cancer. I think 1958, 1959, she ended up dying of cancer. Well, this was one of her pictures. And so she had this picture of DNA, and she hadn't done any work with it yet. And kind of through some backhanded tactics, these 2 guys here, Watson and Crick, they got a hold of that picture. And that picture, you can kind of see an X right here. Kind of see that right there. When they saw that X picture, Watson and Crick, this is Crick right here, this is Watson right here. Crick died several years ago. Like I said, Rosalind Franklin died, I think, the late 50s because of the cancer from the X-rays. James Watson, believe it or not, is actually still alive. And I've heard some interviews from him. He's an interesting character. He's got a unique viewpoint on life, I'll tell you that. But he's still alive. So these 2 guys got a hold of Rosalind Franklin's picture kind of in some unethical ways, and they were the ones that actually published, and they got the Nobel Prize for the structure of DNA. Rosalind Franklin's work wasn't credited because she had passed away, and you can't win a Nobel Prize if you passed away, so it's kind of a discredit to her, but without her work, they wouldn't have gotten to where they did. So I always like to give Rosalind Franklin some credit. She was a female working in a male-dominated field, and she did some miraculous work, and unfortunately, historically, doesn't get any credit. Now later down the road, she's getting the credit that she deserved, which I think is really cool. So these 2 guys published, and then we're not going to get to it today, but this is where we'll end it. It's not escaped our notice, this was in their publication, that the specific pairing that we have theorized or proposed immediately suggests a possible copying mechanism. So these 2 guys stated, like, hey, everybody, here's the structure of DNA. And we also notice, we can kind of see a mechanism for which it's going to copy itself. They wanted the world to know, like, hey, we saw that, but we're not going to publish on that, but we did see it, so at least it's out there. So those 2 guys got credit in history for the discovery of the structure of DNA, and they ended up with the Nobel Prize for it. That's as far as we're going to go today.